In this video, we are going to review some of the terminology we learned in the genetics unit and some of the concepts from that unit. On this slide, we can see the basic structure of a nucleotide. It's very simplified, showing the different subcomponents as shapes. So we have our little circle here, which represents phosphate, our deoxyribose sugar, little pentagon there, and then this little rectangle here represents a nitrogenous base, which would be adenine, which would be pairing with thymine, or cytosine, which would pair with guanine. And on the next slide, we will talk about how they're arranged when they are in the strands of DNA. One of the skills that you need to be able to have is to draw a structure of DNA. So I have it drawn out here. One of the things that you need to know is how to connect them. So if I zoom in here, you can see that the phosphate group from the first one, okay, this was our basic nucleotide structure. So we can see here that it's joined up to the next phosphate at this point. Over here I have shown basically one of those deoxyribose sugars in a little more detail so that you can see where the numbering comes from, the 5' to 3'. So on our more fleshed out carbon ring structure, you can see that the numbering starts over here at this carbon and goes around up to the carbon that's attached up here. So this is the 5' end, and the 3' end would be at the bottom, coming from this carbon here. Other things that you need to be able to label. So you need to be able to label a covalent bond, again showing this one here. Hydrogen bonds, so connecting between the base pairs in the middle, so between adenine and thymine, and again cytosine and guanine. You don't have to know that there is a difference in the number of hydrogen bonds that forms between the different base pairs. That is sufficient just to have dotted lines to show that they are different. You have to indicate where a nucleotide would be. You have to indicate the 5' to 3'. Very important to notice that this strand here is what we consider anti-parallel to the other strand. So you can see our deoxyribose sugar appears to be flipped in comparison to the other strand. So that's something that you need to indicate, and that's really what the 5', 3' will show you as well, which will become more important when you learn about DNA replication and transcription later. To add to the structure of DNA, you also need to know how it actually will organize itself when it's coiling. So this is showing you what we would call a nucleosome, which is made up of 8 of these histones, which are spherical. It's very hard to draw a sphere. And they actually are organized so that the DNA wraps around it. And that will actually help it coil so it can be tighter, so you can fit more information in a smaller space. When we started genetics, we started talking about a lot of different terminology, so I just wanted to review some of that. So we often discuss a chromosome, which is actually discussing a pair of chromatids. So each one of these single structures is a chromatid, whereas a pair of them we call together a chromosome. Often those words are used interchangeably, but that is the more correct definition. And in the middle of a pair of chromatids, or in the middle of a chromosome, we have a centromere, which is what is basically holding the pair together. More terminology that we have related to chromosomes and chromatids are the idea of alleles and genes. So on a pair of chromatids, or on a chromosome, we would find on each of a homologous pair, we would find an allele for a specific gene. So in this case, we could be talking about any gene. Let's call it an imaginary gene that would code for the size of your eyes. In this case, we would have one that has this capital A, which we will talk about dominance and recessive after, and one that has this lowercase a. So the different versions of this eye-size gene are what we would see on each of the chromosomes. So these are separate alleles. The other words that we talked about are the idea of genotype, which is actually when you're looking at the chromosomes and the letters that we would use to code for the different alleles on the chromosomes. So I often say genotypes relate to the letters or the genes, and then the other one is the phenotype, which is actually the physical appearance. So the at the beginning of phenotype and physical go together. So in this case, this individual would have brown eyes. That would be the phenotype of the genotype big B, little b in this case. And the last couple of things that I wanted to review are the idea of dominance and recessive. So whenever we have a capital letter, it usually indicates that it's a dominant gene. So in this example, brown eyes is dominant to blue. So in order to actually have blue eyes, we would have to have 2 copies of that recessive allele. Any other time that we have a big B, the brown eye gene, it would be masking the recessive allele. So if I have big B, automatically brown eyes. And then also when I have big B, little b, we would have brown eyes as well, because that dominant brown gene would mask the recessive blue gene. The other words that come into play in this are homozygous. And when we looked at root words, we talked about homo means the same, and heterozygous. And again, when we talked about root words, we talked about how hetero means different. So in this case, this would be heterozygous because it has 2 different alleles. It has big B, little b. And this example here with the blue eyes is homozygous because it's little b, little b. And then also that big B would be homozygous because, again, it has 2 of the same alleles. And that's your quick genetic review in a nutshell.